In this problem, we are going to classify the enzymes catalyzing those 4 reactions according to the 6 classes. So first is the transformation between succinate and fumarate, catalyzed by fumarate dehydrogenase. So even though we don't know, we are not given the name of the enzyme, but we can analyze what class of enzymes this enzyme belongs to if you take a look and compare the structures of the substrate and the product of the reaction. So what I can see here, that the product, the oxidation state actually of those 2 carbon atoms change, right, upon transformation. In the product, we have -1 for both carbon atoms, while in the reactant, we have -2. So if we have a difference in oxidation states, that we can say that the reaction A is catalyzed by oxidoreductase, because oxidoreductases catalyze oxidation reduction reaction. Now B, what is happening here? So we have fumarate reacting with water to form oxaloacetate. This is another reaction of the citric acid cycle along with the first reaction, which is also part of the citric acid cycle. So we have water here, and we know that if water is used as a nucleophile to break bonds, we have a hydrolase. But here, the bonds are not broken. The bonds are actually made, right? So we have a bond formed between this carbon and OH group and this carbon and hydrogen. So we don't have a hydrolysis reaction, but instead we have bond formation reaction, and we know that another candidate will be a lyase, right? So lyase is catalyzed non-hydrolytic breakage of the bond and formation of the bonds, which we see here. Then C. Again, let's analyze what is happening in the course of this reaction. So we have this phosphate group attached to the first carbon atom of glucose. So it's glucose 1-phosphate converted to glucose 6-phosphate. This phosphate group is actually transferred from the first carbon to the 6 hydroxyl group, right? Hydroxyl group attached to this carbon number 6. So they have a transfer of a functional group, but within one and the same molecule. So if we have such intramolecular transfer reaction, we are actually having an isomerization reaction. So those 2 are constitutional isomers. So the reactant, glucose 1-phosphate, is a constitutional isomer of glucose 6-phosphate. So we deal with isomerase. And in particular, this is a subclass of isomerases called mutase. When we deal with intramolecular transfer of one group into another, we deal with a mutase, which is a type of isomerase. And finally, D. What do we have here? We have the reaction that actually results in the formation of the bond between this carbon atom and this nitrogen atom, right? We have bond formation. And ATP is used as a source of energy. And we have 2 big molecules joining together with the action of this enzyme. So this is a ligase. And this is another enzyme, actually not from the citric acid cycle, but from urea cycle.